Okay, it looks it's working now. So I would like to welcome everyone to this fourth meeting of the South American Initiative in Molecular Simulations, known as SAIMS. And this is a satellite meeting to the IUPAB, to the International Union of Pure and Applied Biophysics Congress that was supposed to happen last year, but due to the pandemic, it's only going on online in 2021. Now, I would like to thank the Congress organization, in particular the people in the SBBf and SBBq societies of biophysics and biochemistry here in Brazil that are also organizing their annual meetings together with the IUPAB Congress. And I'd like to thank all the organizers and the secretary for giving us this opportunity to organize the satellite. Well, I'm not sure if I should give much of a general introduction. I haven't prepared anything, but there's full program. So we are basically having 2 sessions. The first session starting now, we'll have about 11 talks, and so I would like to ask people only to make questions using the question and answer, the Q&A icon in the Zoom platform. And questions can be done during the talks, but the discussion and answers will only be given at the end of each session. That's because we have a small time for each talk, so we think it's better to leave all the discussion to the end of the session. The same thing is happening for the second session that we will start about 4:30, and at the end of it, we're going to have a second discussion. I think that's all the information. Again, I would like to thank everyone, all the participants, all the panelists for being around, for presenting their talks. And I would like to invite Ernesto Caffarena from Oswaldo Cruz Foundation to give the first talk. Oswaldo, please. Can you share your screen? And you are muted. You need to unmute.
Okay.
Okay.
Okay. Hi guys. Thank you very much for the invitation. Thank you, Guilherme, for calling to participate. This is a great opportunity to show what we are doing in our lab. I'm going to show you a video, and I hope no overpass time. Hi everyone, my name is Ernesto Caffarena from I'm with Cruz, Brazil, and my talk is about the process of optimizing a particular peptide named PACAP. PACAP is the acronym for the pituitary adenylate cyclase activating polypeptide, which is a 38 amino acid long peptide similar to the vasoactive intestinal peptide VIP. Both peptides belong to the secreting glucagon superfamily. These peptides work as neurotransmitter and neuromodulators and control cytokine production, cell activation, and cell differentiation. PACAP interacts with 3 G protein coupled receptors that are expressed in various cell types, named PAC1, BPAC1, and BPAC2. These 2 last interact the most with VIP. In studies regarding HIV-1 infection in macrophages, Temeroso et al. In 2013 and 2018 found that cells treated with PACAP and VIP before and after infection showed resistance to viral replication. Also, they observed that they promote mutation in the provirus, reducing thus the viral infectivity. Similar to what happened to HIV-1, it has been observed by the same research group that many patients infected with SARS-CoV-2 presented an exacerbated inflammatory response triggered by the increased activity of the immune system in order to prevent a viral infection. When infected cells migrate and infiltrate the lungs, they stimulate the production of pro-inflammatory mediators. This excessive immune response, along with the unregulated production of cytokines and chemokines, are typical of severe infection with negative clinical progressions in patients with COVID-19. Here, VIP and PACAP can first inhibit replication of SARS-CoV-2 gene in human monocytes and inhibit viral production. Second, they reduce the cellular production of pro-inflammatory mediators in the production of severe tissue damage that result from the infection. And finally, they block the activation of the mechanism involved in its production. When they act together, VIP and PACAP, a synergistic effect can be observed. Because of their consistent anti-inflammatory activities, both neuropeptides have been considered promising therapeutic agents for treating autoimmune and chronic inflammatory disease. In this work, we intend to get new PACAP-derived sequences with greater affinity than the wild type for its receptors to act as agonists or partial agonists and be used for therapeutical purposes. To do this, we use protein engineering in order to develop our PACAP analogs. This technique employs two general strategies: rational protein design and directed evolution. It is based on the assumption that natural sequences can be modified to improve a particular function. For these, 3 points must be taken into consideration. First, we assume that the protein is not an optimum for that function. Second, sequence changes must be done without disrupting the structure, otherwise the resultant protein would not fold. Third, the new sequence must not be too different from the native sequence, otherwise there will be loss in its function. As a consequence, we force point mutation in the protein sequence. Based on the structure of the human PAC1 receptor in complex with PACAP38 deposited in the PDB last year, we built the structure of PACAP in complex with PAC1, VPAC1, and VPAC2. These two last are approximately 40% identical in sequence to PAC1. Then we refine all models and submit them to quality analysis. In the figure at the right corner, we can see that the quality of the models according to the normalized Q-mean score fall in the same range as the crystal available structure fall. Below, molecular dynamics trajectories showed that new interactions appear and some are maintained, while others are lost. Red dots indicate interactions between PACAP and PAC1 that only appear in the crystal structure and were not maintained in dynamics. The yellow ones represent the upsurge interactions, while the green ones denote those seen in the simulation and the crystal. In the figure at the left bottom corner, the interaction energy between PACAP and receptors is displayed. Sorry. Looking at the peaks, data corroborate a better interaction energy between PACAP and PAC1, agreeing with affinity experimental data, although only the enthalpy contribution was taken into account. Aiming to get an agonist or partial agonist from PACAP, we performed computational analysis, alanine scanning, only on the first 15 residues of PACAP38, which are those that are inserted into the receptor and contact it directly. We employed MMPBSA for the free energy calculations. Residues asparagine 3, asparagine 8, arginine 12, and arginine 15 were detected as hotspots. They are critical for triggering biological responses. On the other hand, outcomes from the network analysis showed that these identical residues presented high centrality measures, the so-called hubs, reinforcing their essentiality. So these 4 residues remain unaltered. Despite being described as a critical residue, alanine scan results showed a slight positive variation of histidine 1, so we decided to include it in the mutation analysis. On the lower left corner, we can appreciate results derived from molecular dynamics and network correlation. Again, several interacting clusters were detected, one for each color, where matching colors in the peptide and receptors denote high interaction. Residue mutations were performed using two scripts from Rosetta package. We followed different approaches. When we regarded only the absolute binding energy, the wild type that was retrieved as the best sequence was the wild type, showing that the nature is somehow unbeatable. So it was necessary to improve the strategy by adding some restraints, such as including the thermodynamic cycle and conserving the secondary structure with an interfacial structure RMSD less than 2 angstroms. We used two sets of structures as inputs, one composed of structures derived from refined models, and the second composed of the most representative configuration from MD simulation. In the sequence shown in the slide, only residues in red were allowed to mutate. The figure below shows the secondary structure corresponding to wild type that was taken as a reference. Of a total of 3,000 redundant sequences, we focus on the region demarked by the red square, where negative delta G and IRMSD below 2 angstroms corresponding to the sequence with a minor secondary structure deformation were located. Sequences derived from the refined structure resulted in lower delta G values with better score, while the set derived from the representative configurations resulted in the greater diversification. After eliminating redundancy and applying these filters, only 57 were regarded for further analysis. Finally, here we have the comparison of the secondary structure of the wild type and the analogous sequence. And for a question of security sakes, I can show the sequences. Finally, to sum up, a SWOT diagram shows the main topics related to this project. As a strength, we highlight the computational pipeline was capable of generating a considerable volume of mutated sequences. As a weakness, we remarked that computational cost due to the number of several MD simulations performed, and second, the existence of plenty of false positive sequences. Opportunities appear when this methodology can be applied to other secretine family members, such as glucagon. One fantastic point is the possibility of obtaining patents. And finally, as a threat, we mentioned that we are in the test stage right now. And finally, I want to thank my team, which is composed of Fernando Meirelles, Deborah Antunes, Dumich, Bob Habib, and Jairo Timeroso, and the agencies SBBq and CAPS. Thank you for your attention.
Okay, thank you, Ernesto, for your talk. As I mentioned, we can have questions typed in a Q&A, and those will be answered by the end of the session. So we better move to our next speaker, who is going to be Diniz Sena Jr. from the Universidade Regional do Cariri. Diniz, can you share your screen, please? We can see it, but we cannot.
Hello, everyone, and good afternoon. It is a pleasure to be here at the 4th meeting of the South American Initiative in Molecular Simulations to present my talk, A Closer Look at the Mu-Opioid Receptor, an Enhanced Sampling Approach. My name is Diniz, and I'm a professor at Universidade Regional do Cariri, which is located in the south part of Ceará state in the northeast region of Brazil. GPCRs are very interesting proteins which transfer information from the outside to the inside of the cell by interactions with ligands, which can be peptides, hormones, and even light. And as such, they are the target of more than 30% of drugs in the market. They share a common structure comprised of 7 transmembrane helices connected with intra- and extracellular loops. I am particularly interested in the mu-opioid receptor, which is a class A GPCR and the main target of morphine. After activation, conformational changes take place that will trigger different signaling pathways. And as few experimental structures are available, whole-atom molecular simulations fill this gap to help interpret the activation mechanism. The available structures of the mu-opioid receptor are corresponding to the inactive and to the active form of the protein. The inactive form was published in 2012 under the PDB code 4DKL, while the active form was published in 2015 under the PDB code 5C1M. In 2018, another structure was also released in the active form of the receptor bound to the G protein. All these structures are from the murine receptor, from the rat. However, they share 94% sequence similarity with the human receptor. And so they are suitable models to be studied. We can see the difference between the active and inactive form of the receptor in this picture. Looking from the intracellular view, we see that the transmembrane helix 6 suffers a large splint after activation of the receptor. There are also other features that are observed on receptor activation, some molecular switches, some motifs that are also observed. But this is the most striking difference between the active and inactive form. And it is common also to call the active form the open form and the inactive form the closed form of the receptor. After activation, the mu-opioid receptor may signal through different signaling pathways. One of them is the G protein pathway, which is associated with the pain-killing effect of morphine, and the other pathway is the beta-arrestin pathway, which is related to the undesired side effects of opioid drugs, like respiratory depression, addiction, constipation, and so on. So we are interested in our simulations to observe the transition between the active and inactive states of the receptor. It is known that a mutation in residue 150, which is an asparagine or an alanine, turns the receptor into a constitutively active mutant. That is, this mutant will be active even without the presence of a ligand. We performed some molecular dynamic simulations using GROMACS 4.6 with the Amber 99SB IRDN force field with an adaptation to include parameters from the Stockholm lipids. In the classical molecular dynamic simulations, the wild-type complexes and the APO receptor were used, as well as the constitutively active mutant. After simulations of hundreds of nanoseconds long and even microseconds, we were not able to observe any transition from the active to the inactive conformation. Then we decided to apply an enhanced sampling technique called REST2, using the APO receptor in the constitutively mutant form, and also some wild-type complexes. Using this scheme, we prepared 64 replicas, and each one was run for 20 nanoseconds each, comprising a total of 1.28 microsecond simulation time for each system. In the replica exchange with solute tempering, the REST2, we adapt the force field parameters so that they scale in order to simulate higher temperatures. This is done for the receptor and the ligand. The rest of the system remains untouched. After a number of steps, replicas with similar energies are exchanged, but only the first replica is analyzed. In this figure, we can see the evolution of 4 replicas being run simultaneously, and from time to time the structures from one replica are exchanged with the structure from another one. If the energy differences between these structures is not suitable, then the exchange will be forbidden. For the apoproteins, the wild type and the constitutively active mutant, we observed some interesting results for the seconds. There were some transitions whose structures were observed and compared with the available active state structure of the receptor. These simulations were run on the Jülich Supercomputing Center in Germany, and we can see here that the transmembrane helix 6 from our simulated structure overlaps very well with the structure from the experimental crystal. The results were published in this paper on Scientific Reports, and you can access it using the QR code here on the right. Then we decided to perform the same simulations using the REST2 scheme with the wild-type receptor in the inactive and the active form. For the active form, we used the original ligand BU72, while for the inactive form, we used morphine, which is also an agonist as BU72, naloxone, which is an antagonist, and naloxone benzoyl hydrazone, which is a partial agonist. These complexes were embedded in a POPC membrane and hydrated with water and sodium chloride up to a concentration of 0.15 molar. And these simulations were carried on Santos Dumont supercomputer at LNCC in Brazil. The results of the research were published in the Bioquímica and Biofísica Acta General Subjects, where you can access using this QR code here on the right. Some of the results are shown here. We can see in this figure the green ribbons represent the experimental structure of the active conformation of the mi-opioid receptor, while the gray ribbons represent structures from our simulations. We observed the transition from the conformation of transmembrane helix 5, but not from transmembrane helix 6. However, we have also observed some other features which are different for agonist and antagonist in these simulations. Morphine, for example, increased the distance from the residue R165 and T279, while naloxone, which is an antagonist, increased the RMSD of the amino acid motif NPXXY. So these are different behaviors from the agonist and antagonist which were observed in these simulations. So we were very happy to use the REST2 scheme for this system because it allowed us to see features that were not able to be captured on classical molecular dynamics. And the structural similarity between GPCRs suggests that these methods should be readily transferable to other systems as well. And data analysis should take into account the lack of time sequence continuity because of the exchanges along the simulations, and principal component analysis and unsupervised clustering are very helpful to analyze this kind of data. So this is my current group in Lazemol. Mira is working on her PhD. Josué has finished his master thesis. Guilherme, Ruben, Mariana, and David are undergraduate students all working on the mi-opioid receptor. So thank you very much for your attention, and now I am open for some questions.
Okay, thanks a lot, Diniz. Thanks for your talk. And I think we can move to our next speaker. Now, Esteban Martinez from Universidade Concepción in Chile. Esteban, can you please
Yes.
share your screen?
I will try. Give me 1 second.
Are you in Chile or in San Francisco?
No, I'm in Chile, actually. This is the wrong picture. I don't know why. So I will try, and I hope you can see my slide. I will do it the old way. Can you see the presentation now?
Yes.
Moving forward? Okay, great. So thank you very much for the nice introduction and the invitation. Today, I want to show work done by 2 PhD students, Juan Gonzalez and Luis Macaya, who are working in my group. The group is the Quantum Chemistry Molecular Modeling group in Universidad de Concepción in Chile. And we are looking at phosphate parameters to improve basically the interactions in biological systems. And I will just start with the introduction. So our goal basically is to predict receptor ligand binding affinity by computational affordable methods. That means that we want to be accurate and at the same time fast. Why? I know I don't have to explain it to this audience, but basically we want to contribute to new methods to speed up small molecule drug discovery. And our focus is basically to improve the description of molecular interactions and use advanced sampling algorithms with respect to the chemical coupling parameter lambda, using, for example, the self-adjusted mixture sampling method. So for the first step, we looked at host-guest systems because these systems exhibit the same non-covalent interaction as proteins and don't suffer from the sampling issue, which is sometimes difficult to address. That means that we get converged, or we can get converged results within 2 to 3 days on one GPU for one system. And the other, I think, important thing is that they have well-defined protonation states that so that at the end, the binding free energy we get out depend only on the force field and not on other parameters. At least we have validated that before. We basically looked at 3 different hosts to our octa acid host, which differ only in having methyl groups in the upper opening, and the CB7 hosts, which are all well characterized apart from benchmark systems in the computation in the community. So let me just jump to the results. So I will just show the results going over each host. I will start with the Timoa host. This host has 4 methyl groups in the upper opening. That means that water molecules have difficulty to reach or enter the host. And what I show here are basically the 6 guests which we studied. And the graph shows basically the difference between our calculated value and the experimental value expressed in kcal per mole. And we compare basically using the standard, and maybe most of you are familiar, GAF AM1 BCC atomic charges, and using then the charges. Actually, there's something missing here, methodology. Sorry, I missed this slide. I have to explain first one thing. Sorry for that. So let me just introduce the methodology because this slide somehow got lost. So what we do basically in difference to normal free energy calculations is that we derive the atomic charges and the non-bonded force field parameters direct from QMMM methodology. That means we run QMMM calculations, we take the polarized electron density and derive then this non-bonded force field parameters using a partitioning method, which is called minimal basis iterative Stockholder method. We have written a Python interface which works together with the Orca quantum chemistry package and the OpenMM simulation package, and basically derives the atomic charges for the ligand. And we also calculate polarization energies, which contribute significantly then to the binding free energy. To calculate then the binding free energy, basically, we use the Young software by the Codera group, which is very efficient, and we apply standard alchemical protocols where we decouple basically the interactions of the guest or ligand in the solvent and then bound to the host. That is important so that you get the next slide. So now what I compare here is basically our atomic charges, which we derive, as I have shown you before, to the AM1 BCC charges, which is most people use for ligand, protein ligand binding. And basically the error to the experimental value is shown here for each atomic charge, and you can see for each different guest. And in most cases, basically the one in violet, which are the DM-BIS charges we use, are smaller. Only in some cases we have larger errors, mainly in the ones who have ammonium group, where we have to look into a little bit how the electrostatics are basically provided there. If you now compare, this is the absolute value. If you then look at other statistical descriptors, as for example, the RMSE or the MIE, we also, our charges reveal a smaller value. So that means they match better to experimental value. Also, the correlation coefficient is better. And importance, this Kendall-Tau, maybe some of you don't know it, but Kendall-Tau basically describes how well you can order the guests from strong to weak binders. One means you order them correctly, minus one completely the other way around. And basically what we can get with our charges is that we order them correctly according to which of them are strong and which of them are weak binders. If we switch now to the other host, the octa-acid host without the methyl groups, that means there are more water molecules which can be then in the host. We also see that, again, our charges now derive for the host and the guest. In violet, the errors are significantly smaller than the one in AM1 BCC. This is the absolute value for the same guests. And the statistical descriptors are also smaller. That means we are closer to experiment. We go from 2.6 to 1.8. And also the other values basically show a better agreement with experimental values. So we were quite happy. So we switch now to the next host, the CB7. This host is different from the other ones because it is a polar host, but it doesn't have a net charge, so it's uncharged. And these are the guests which were studied experimentally with this curcubit uryl host. And again, basically, we have small errors to experimental value using our charges, these EM-BIS charges, compared to the AM1 BCC charges, which in some cases have considerable error. Again, the RMSE value also drops, and we have also kind of a better correlation, especially the Kendall-Tau. So again, the ordering from weak to strong binders is better with our charges. These are all unpublished results, the CB7 results. So that made us quite happy. So we moved on, and then we looked at this host, which have adamantine cores, and now things doesn't work very well, because basically our violet bars are all larger than the green ones, and this is sometimes how science works. So sometimes you find that things get worse, although you expect them to get better. So we asked what are we missing? And we looked at the interactions of this host, a guest to the host, and we found basically that this host, because they have this adamantine core, they are governed by exchange and dispersion interactions. So we went back and recalculated the Lennard-Jones parameters for these guests shown here with our methodology, and these are shown in gray bars here. And now basically our errors drop significantly compared to the AM1 BCC charge and to the ones I showed you before, so the violet ones. So that means that by using our methodology to get the atomic charges and the Lennard-Jones parameters, we get better agreement with experiment, and also the statistical descriptors. So the root mean square error and also the absolute error, they are almost half, and the coefficient and Kendall-Tau, I would say, they are almost the same. So with that, I'd like to conclude. So basically, I hope I could show you that non-bonded force field parameters derived from MBIS, partition polarized molecular electron density, improve the binding free energy prediction in host gas systems. And so this is not working properly. Sorry for that. The new implemented workflow to obtain the parameters is almost automatic, starting from an undercut topology. And as references, so the results about the octa-acid host were recently published in the Journal of Chemical Information Modeling, and the results in CB7 are unpublished, will be submitted soon to the same journal, and I hope. And the software we use to obtain these parameters, they are available at the group GitHub page. This is already available and for download, and you can try it out. And the new one, which will come out until in the next of the year, the end of the year, is the one over here, and that will become available. So that, thank you very much for your attention, and I open for questions after the session. Thanks.
Okay, thank you, Esteban. Thanks for your talk and your participation. We're going to move to our next speaker, who is Bruno Horta from Federal University, or Universidade Federal do Rio de Janeiro. Bruno, can you please share your screen? And unmute your microphone.
Do you hear me?
Yes, I do.
Okay.
You can start.
I'm having some issues here with my screen. Let's see. Do you see my screen?
Yes.
Okay. So first of all, thanks a lot, Pilar, for pushing this forward and organizing this meeting. It's very nice to see all of you here today. So I'll put a hard stop here on my alarm clock just to not exceed the 10 minutes time. I'll talk today about our recent efforts in automatic force field parameterization. So in our lab, we have a few projects about software development. So some of them are focused on topology building, like the PyPoly builder that we recently published, and also for helping the development of new force fields. So the work I will present today is the PhD thesis, part of the PhD thesis of Ian Gonçalves. And this work is divided into two main areas. Probably I won't have time to talk about the first one. But before that, why we automatize, right? So it's important to think about automation, not only in terms of saving time and human effort, but also in terms of reducing the non-systematic errors that may occur, and also to be more reproducible and to explore more the high-performance computing facilities that we have access to. So in this regard, we are developing two different softwares in our lab. Both are part of Ian's thesis. One is the grid maker, as we call it today, is for helping with optimizing the non-bounded parameters for condensed phase systems. And the other project is to help developing new torsional terms that are also coupled with 1,4 interactions. So in this last case, I'll probably not have time to talk about it, but just to briefly explain, we have this simultaneous optimization of both the torsions and the 1,4 terms. And also it's a very flexible program that you can have the optimization of multiple torsions and also define all the systems that are going to be the target for this optimization. I'll try to focus more today on the other project, that's the grid maker, that helps to automatize the parameterization of both charges and Lennard-Jones parameters. So we call it GMAC. This is a customizable driver for force field parameterization. What do I mean with customizable driver? We try to make it as flexible as possible to accept different softwares and also different systems, different properties. So the main aspects is that we discretize the parameter search space, okay? And we use surrogate models to estimate what is the effect of the change in one or more parameters. So, and we control the statistical uncertainties by automatically extending the simulations when needed, because to have a good estimation of the effect of changing some parameter, we have to take into account also that the simulations have to be statistically sampled with enough for enough time, so we can better estimate these properties. Also, we have iterative updates of the search space boundaries. So if the discretized space that you initialize your system is not enough, the system automatically expands the search space and walks along the direction of optimal parameters. And it's completely modular and customizable. So I'll just explain how it works. This is just an example of optimizing a pair of C6 and C12 parameters. So we would discretize the grid. Some points will be selected for actual simulations. So in this case here, we are simulating 9 different points. All the other points are estimated based on a given surrogate model that could be either M-bar or interpolation methods or machine learning methods. Then the topologies are created, the systems are simulated or extended, the properties are calculated and filtered and treated statistically, and we analyze for convergence. If it's not converged, then we calculate how the new length of the extension of this simulation, and we extend the simulation. If it's converged, we estimate the properties for all the cells in the system. Then we estimate some scores of quality. We have some thresholds that we have to respect, and then we decide if we move or not the grid. If not, then it's done. If yes, then we have to move the grid in the direction of the optimal parameters. So basically that's the approach. So this flowchart can be customized by the user. So we have basically 2 levels of customization. So the first level of customization, we call it soft level, is the level where you can specify all the parameters for different classes and the methods that are already implemented in the software. So for example, we have implemented machine learning algorithm and also interpolation method, and then you can choose either you do with interpolation or with machine learning. That's an example of soft customization process. But the second level of customization is what we call the deep customization level, where you can replace or add new methods that were not previously implemented in the code. You can add your own methods. Let's say you like to do that with M-bar, for instance, which is not implemented yet. Then you could implement your M-bar estimator and use your M-bar estimator. So the soft customization is done in the input level. So we have an input file, so you can define all the procedures. And the deep customization is done in 2 different steps. So you define a new object, which is done using an API that we provide. So you add, for example, a custom surrogate model, or you add a new scoring function. You can do that, etc. And reference to this new object in the input file by the name that you baptized it in your new object file. So we'll give some illustrative examples. This is a simulation. Okay, for simulation, we usually use GROMACS software. I'm actually running out of time. I'll have to speed up. But just to say, we have implemented it with GROMACS, but you can implement your own function to use another software, for instance. That can be done. The workflow functions with dictionaries. So you specify, for example, what are your output files, etc., and you can quickly set up that. There is a tutorial for doing that. I will go very quickly here now because really I am almost ahead of time, but I'll show you some results. This is a master thesis of a student in my group, Ricardo, that is using the grid maker to optimize a methanol model. This is the type of results that we get. So we enter with the atom type. We choose to optimize, for example, the C6 and the C12 parameters of the oxygen atom of methanol. And then the program, the grid maker finds the optimal solution. Here is just the final data for rigid model and for flexible model, respecting 3 different properties. So our 3 target properties here are the density, the heat of vaporization, and the surface tension. So it's just to illustrate what kind of results we get. And of course, since we have many properties being optimized, we cannot find a unique solution for this problem, but rather we find a Pareto frontier. So in this case, you have to select which is the, and I am almost out of time here. You can select which is the optimal solution that you prefer. So here on the left, I show you the solution space in terms of the parameter space. All the points in orange are optimal in the Pareto front. And on the right, in terms of the objective function space. So there are several points there that are the optimals in terms of the heat of vaporization, the density, and also the surface tension. So let me quickly move to the last slide of this presentation, just to thank you all for your attention, and also my group. So as I mentioned, this is the work by Ian Gonçalves, and also the application work by Ricardo Monserrat. Thank you so much.
Okay, great. Thanks a lot, Bruno, for your talk and your participation. I would like to thank all speakers that have been trying their best to be on time. I really appreciate that. We have a lot of speakers today, so we have to be tight in the time schedule. The next speaker will be Darío Estrin from the Universidade de Buenos Aires in Argentina. Darío, please, can you share your screen? And unmute your microphone. Yeah, Darío, we can see your screen, but we cannot listen to it. Okay, we can now. Hello?
Oh, okay.
It's working.
So
yeah.
can you see my screen?
Yes, it's fine. Carry on.
Okay, so I'll start. Okay, so first of all, I would like to thank Guilherme for the invitation, for having gathered all these people in this nice meeting. I hope that next time we can do it presentially. I'm going to talk about work that we have been doing in collaboration with people from Uruguay, from the University of the República. And it's a combination of experiments and simulations, basically regarding a very interesting kind of species that are called persulfides. Persulfides are species that are analogs to peroxides, but with sulfur atoms, and are very important species because they are involved in the metabolic pathways of sulfur, and they are proposed to mediate the signaling effects of hydrogen sulfide. Hydrogen sulfide has been found in the last years to be very important in signaling. It's another signaling molecule, more or less in the same condition as nitric oxide or carbon monoxide, and their chemical properties of the persulfides are not completely understood because they are really elusive species. And so we propose to study the formation, the acidity, and the reactivity of these species by using experiments with the glutathione persulfide, because this is very abundant physiologically. And We compare the results with calculations performed with glutathione persulfide, and also we perform calculations with a model methyl thiol and methyl persulfide, because they are smaller molecules, and that allows to make more extensive calculations. And so the idea is to try to understand if these species compare the persulfide with the thiols. The intrinsic reactivity of these species is due to the reactivity of the anions, because the anions are involved in the nucleophilic reactions. And so it's very important to understand both the acidity, to see the amount of the protonated species at physiological pH, and on the other hand, the reactivity, the intrinsic reactivity of the anionic species. And we investigated this reactivity towards different targets: hydrogen peroxide, peroxynitrous acid, that are physiologically relevant targets, and another target that is monobromobiman, that it's a target related to analytical investigation of this species. It's a molecule that is used to characterize this species. And so we resorted to multi-scale calculations to shed light on this issue, and we use the multi-scale calculations in different ways. We started by performing QM calculations at different levels of theory to validate the methodology, to see which kind of methodology has to be used to extract meaningful results. We have also performed classical molecular simulations to understand the complexity of the potential energy surface in aqueous solution. And at the end, we finished by performing QMMM calculations using our code, the Leo code, that is a DFT code that run on GPU and is very efficient. And all these calculations, we have performed the calculations in intimate collaboration with the experimental group. And so we started with the acidity. The acidity is a very difficult issue. If you decompose the deprotonation process in different chemical steps, we can see that you have very exergonic steps and very endergonic steps. The bond breaking steps are very endergonic, and the solvation steps are very exergonic. And so it's a very difficult issue, because the net result is very small, and it's due to the compensation of large negative and positive numbers. And so in this way, we have to resort to different kinds of calculations using continuum solvent model calculations, using different levels of theory, of electronic structure theory. And we see that you see dispersion in the results, because there are small errors or small variations in the computed quantities lead to differences in the global process. But anyway, we have been able to extract trends. And the trends are that the persulfide are more acidic than the thiol, than the corresponding thiol. And besides that, we saw that the main source of difference is the bond breaking of the sulfur-hydrogen bond. Because the relationship between the pKa and the delta G is exponential, small errors in the delta G lead to very large differences in the pKa. And the experimental pKa of persulfide is about 5. This is a value obtained by the people in Montevideo. This is the pKa obtained for the first time. And we obtain a difference in pKa of 7, while the experimental value is about 3. The experimental value says that the persulfide is 3 units of pKa more acidic than the thiol, and even at coupled cluster level, the result that we obtain is not quantitatively correct, but anyway, the trends are correct. Well, we went to the nucleophilic reactivity. The experiments have been performed by running stop flow experiments. These are very difficult experiments because the reactions are fast and you have to be very careful. And so at Montevideo, they perform the experiments at different temperatures in order to extract delta G of activations from the kinetic results. And we perform the simulations again at the first time by using PCM calculations in combination with several levels of theory. And in the second time, we perform the calculations by using a QMMM umbrella sampling approach. And so we were able to compare the more accurate regarding to the electronic structure method, but with more simple continuum model approach, with more sophisticated QMMM calculations that allow us to consider the solvent in an explicit way, but we are restricted to use just one DFT scheme because the QMMM calculations are much more expensive. And so we see from the electronic structure calculations that the delta G of activation between the thiol and the persulfide are very small, and they are also dependent on the target. We obtain this by analyzing overall the different electronic structure calculations, and by using the free energy approach, by using QMMM simulations, we were able to perform QMMM molecular dynamic simulations. The reactions are SN2 reactions, so the reaction coordinate is very simple, and we were able to obtain these free energy profiles, and the free energy profiles confirm the same message that we obtained before, that the intrinsic reactivity is more or less the same, and the same conclusion can be obtained with the experimental values. You see that the delta G difference is really close to the experimental value. I'm not going to talk about the solvation patterns because I'm running out of time, but the take-home message is that the persulfides are much more acidic than the thiols, and they are completely deprotonated at physiological pH for these physiologically relevant persulfides, and that the intrinsic reactivity is not very different. So the enhanced reactivity that is observed physiologically is mainly due to the fact that since the reactive species is more acidic, the reactive fraction, the deprotonated fraction is higher. And the other message is that the combination of experiments and simulations is very useful to tackle this kind of complex chemistry. These results have been published recently in a JBC paper, and are the results obtained by Jonathan Semelak, who performed the simulations, and Diana Benchoam in Montevideo that performed the experiments. Thank you very much.
Okay, great. Thanks a lot, Darío, for your talk and participation in the meeting. I will move to our next speaker, who is going to be Hugo Verli from the Federal University of Rio Grande do Sul, Universidade Federal do Rio Grande do Sul in Brazil. So, Hugo, can you...
Good evening to everyone. My name is Hugo Verli, and I would like to thank SBBq, IUPAB, and Guilherme Arantes for the organization of the symposium. Today, I would like to present to you some results we obtained recently regarding the current status of carbohydrate structure information on PDB, and how these results had inspired us to advance in the calibration of force field parameterization for carbohydrates. Carbohydrates are known to possess a large potential chemical information content, which is a consequence of the chemical diversity we found in nature on the monosaccharide level and also on the glycosidic linkage level. This high diversity of potential scaffolds formed by carbohydrates implies in a huge plethora of biological functions. We find carbohydrates interfering in protein function, dynamics, folding, allosterism, catalysis upon glycosylation, but we also find carbohydrates participating in molecule-molecule recognition, cell-cell recognition as biomarkers for several diseases, participating with key roles in inflammation, immunology, and so on. This plethora of biological roles open several avenues for investigation towards drug development. Unfortunately, these efforts are highly impaired by the low amount of structural information regarding carbohydrates on the PDB. Additionally, to the low amount of structural information, there are several problems recognized with these entries, such as problems with stereochemistry, naming, residue identification, resolution of the electron density in general. So we implemented a routine that intended to circumvent part of these problems, offering a clear identification of the monosaccharides and filtering for quality the entries. Accordingly, we observe that about 2/3 of the PDB possess information of medium to low quality. This low amount of structural information is even additionally concentrated on a small set of monosaccharide residues. Finally, the information about this small set of residues is highly concentrated at the monosaccharide level. We have about 1/3 of the information, crystallographic information on PDB about carbohydrates containing data about glycosidic linkage, which is pivotal for building polysaccharide structures and validating molecular modeling models. This glycosidic linkage information on PDB is additionally concentrated on beta-1,4 linkages, particularly on beta-glucanac linkages. As a consequence, we may see that the current level of information of carbohydrate information on PDB severely limits the insights into search for insights into the biological roles, impairing further investigations on the biological function of most type of carbohydrate residues, and also impairing the building of models for polysaccharides based on experimental data. One additional confirmation aspect that is particular for carbohydrates is the puckering of exopyronosis. This puckering was observed in PDB to increase up to 6 times upon the lowering in resolution. So the ring distortion increased severely in medium to low resolution structures, which may have a large influence in the interpretation of the biological roles, biological function of these distortions. It could be in several situations, in most of the situations, just an artifact from experimental conditions. But still, we found around 10% of the residues distorted in high resolution structures, which may implies that these residues are in fact distorted by physiological conditions. In several situations, like in glycosidase, previous authors indicated that these distorted conformations are a pathway during catalysis that were trapped in the crystal structure. And in fact, we see that the residues, each dot is a residue in the PDB. Some residues are located on local minima like bolts or skew bolts, and also on transitional states. But we see a lot of points in high energy structures, in high energy regions of the map, inverted chairs, and in structures that are not enzymes, like proteins. And we don't have, in most scenarios, a clear explanation for these situations. One approach that is commonly used to explore these processes in nature include the use of enhanced sample techniques like metadynamics. These are two examples for the exploration of the free energy landscape along the theta angle for the free residue in red and for the complexity residue in the dotted lines, here for glucose and here for mannose against the same enzyme. In stars, we see the experimental geometry, and we clearly observe that in some situations the enzyme is able to interfere in this free energy landscape. This obviously is limited by the accuracy of the employed force field, and the problem with using QMMM techniques is the limited sampling we can obtain for the protein in the systems. So the development of force fields able to better describe this process could aid in higher sampling and a better exploration of the dynamics of this molecular recognition and the biological processes implied. About 10 years ago, we developed a set of force field parameters for carbohydrates based on Roberto Lin's parameters for some exopyranoses under Gromos. So we expanded with him these parameters for most of the exopyranoses found on biological systems, including parameters to all the glucosidic linkage type known between carbohydrates and proteins. We validated these parameters in several papers, and just to exemplify, we can see in black dots the experimental geometries for this specific glycosidic linkage, and behind the free energy landscape for the linkage, indicating that the experimental dots are well within the theoretical maps. Unfortunately, Gromos is known to overstabilize the chair forms, so we have a poor description of intermediate conformation and of the conformational transitions between the chairs. So we are now expanding these parameters for in this case, the family of sialic acids, now considering the reproduction of vicinal coupling constants in water. In this case, we can see for two anomers of this neuraminic acid a very good reproduction of the ring conformations. In some more nasty situations, we can see carbohydrates fused to aromatic rings in natural products, and the presence of some boat conformations. And we have experimental data for the presence of these both conformations, both from crystal structures and NMR data, and after refinement of the torsional terms in these rings, we can see now on unbiased simulations the presence of these boats. We are moving now to a fine calibration of multiple residues using as the fitness function the vicinal coupling constants, and we can see in this case for those an excellent reproduction of NMR data. Under multi-microsecond simulations, we can see the conformational transitions between the chairs arriving to relative abundance of the two chairs very close to those expected in solution. The equilibration of these parameters are quite slow, but they offer at the end an excellent fit to the experimental results. Unfortunately, this should be done to each residue individually, so we are now adjusting for several residues the torsional terms in order to offer to the community better parameters to solidly reproduce sugar ensemble, and then return to those studies of the carbohydrate within the protein. I would like to thank the students and collaborators that participate in these results and the funding agents. Thank you very much for your attention.
Okay, thanks a lot, Hugo, for your talk and your participation. Our next speaker will be Lucas Bleicher from the Universidade Federal de Minas Gerais. Please, Lucas, help yourself. Can you unmute and mute, sorry.
Okay, thanks.
Okay.
So I talk a little bit about how to do research with computational biology and evolution. In the last few years, I have been working on how to extract information on multiple alignments, which tend to have a lot of evolutionary data embedded. And so they represent homologous sequences which derive from a common ancestor, and also the evolutionary restraints. So of course, when we see residue conservation, they are very commonly related to aspects of structure and function. For this example, these positions are related to catalytic residues. What we have been doing is to try to extract further information from coevolution data. So the idea that when we see positions in a multiple sequence alignment that tend to correlate with each other, this can also be related to even more interesting information on structure and function. And as you all probably know, we now got to a point on which this kind of information is now used very commonly, even for ab initio protein structure determination, because we now can derive methods to predict contacts from co-evolved positions. But instead, we also try to find other types of useful data from the coevolution studies, and the fact that when we see sets of positions which co-evolve, they very commonly are related to functional sites. So the basic idea is that we start from a multiple sequence alignment like this, and then we find patterns of positions which tend to be correlated to each other. We create a network which will represent this information, and of course they will always be much more complex than this one, but then we can decompose this network into sets of correlated positions which generally are reporters of functional sites or subclass determinants in protein families. Okay, well, I can. So we have been using this for the determination of function for novel proteins, for gene annotation, because usually finding these sets of co-evolved positions and then looking at the literature, we are able to predict what reses are related to given characteristics in protein families. So we have been using this for novel protein functional determination, and also for the rational design of mutagenesis experiments. This is something that we've been doing with collaboration with other labs, but also in silico mutagenesis. So this is something which, if I remember correctly, I included in my last presentation for SAIMS. We have published this, for example, in which we derived molecular simulations studies by looking directly at which residues pinpointed by this technique are likely to seriously affect the structure and functions of a given protein. But what we have also been doing is looking at the evolution of novel functions. So basically, it's common that once there's a gene duplication followed by divergence, a lot of mutations can accumulate and generate completely different proteins which are still homologous to the older one. And we've been studying this set of proteins, the HIUAses, which are enzymes in the uric acid degradation pathway. They are present in all kingdoms, but during the evolution of vertebrates, there was a gene duplication, which gave rise to another protein, which is even more known than it, which are the transthyreteins, which is a classic model for studying formation of amyloid fibrils. And they are the main hormone distributor, thyroid hormone distributor in the brain. And so they were generated by duplication of HIUAses, then further modification. And we have decided to study these proteins using the technique of ancestral sequence reconstruction, which is a statistical inference of internal nodes on an evolutionary tree. We have access to modern proteins. We can generate a phylogeny, and then we can calculate what would be the most likely sequence for proteins that existed many million years ago. The theory for doing that is not recent. Linus Pauling and Zuckerland developed it still in the 60s, but only recently it was able to be done experimentally. Now you can just synthesize the DNA sequence from a commercial source and study those proteins. So we did that to the ancestors of transthyretines and HIUAs. So we did experimentally with X-ray crystallography and ITC. So if we look at the protein, the most likely protein just before gene duplication, we saw that it wasn't able to bind to the thyroid hormone. But when we generated the sequence of the protein, which most likely sequence just after gene duplications with just a few modifications in amino acids, it was already able to bind T4, the thyroid hormone, as can be seen in the crystal structure, and also in the ITC experiments. So it's not too different in terms of the dissociation constant to the modern human transthyretin. And what is interesting is that when we compare the mining mode for human transthyretin and this ancestor that originated more than 500 million years ago, we have basically the same binding mode for the thyroid hormone. It was virtually unchanged since then. Of course, we would also like to study the catalytic activity aspect of the older protein in this family, the HIUAs, which was what they did before gene duplication. And so we use molecular docking to compare the binding mode of the HIU, which is the substrate, and also the high energy intermediate when converted to OHCU, which is the product of this reaction in HIUAs. So we compared it with a modern HIUAs, the HIUAs transthyretin ancestor, which we calculated, and also the transthyretin ancestor, which was right after gene duplication. So it was what we could see is that the binding mode for the ancestor, which even older than the duplication, was very similar to the modern HIUAs, but completely incompatible with transthyretins. There are some other stuff we would like to do with this system, which is very interesting to study using both computational and experimental methods. For obvious reasons, we have not been able to do experiments a lot in this last 1.5 years, but we'd love to do enzymatic assays once we are safe to go back to the lab. And I'm saying all this because I'm also looking for collaborators. We would be very interested in study this reaction of conversion of 5-hydroxyisourate to HCU using QMMM, and then compare how do they relate when we compare the modern HIUAs in this ancestral HIUAs. So that's what I had to say today, and thanks a lot for the opportunity.
Okay, thanks, Lucas, for your talk and participation in the meeting. I just would like to mention that at the moment we have more than 90 attendees watching this. So I'm really happy that we had a good turnout of people. And I'll move to our next speaker, who is going to be Pilar from the Universidade de Antioquia. in Colombia. Pilar, can you please share your screen?
Yeah, so good afternoon, everybody. I'm very happy to be here. I would first like to thank the organizers for this great invitation. I hope next time we could do it in person. And today I'm going to talk to you about peptide design towards the major histocompatibility class 2. And this is a work done by my PhD student at the University of Antioquia. So can you see my next slide? Okay, so we're going to, the idea is to design peptides that bind with higher affinity to the MHC class 2 receptor. This is a receptor that grabs exogenous peptides and displays them on the cell surface and then triggers immune response. So it is one of the key pieces in the immune response pathway. And so this is towards synthetic vaccine design and synthetic T-helper design and adjuvant design. So basically anything that boosts the immune system. So this is a protocol, the computational protocol that we've developed. The idea starts with an initial structure of the receptor, the MHC class 2, bound to a peptide. And then what we do is we randomly pick a place on the peptide chain. So one of the positions on the peptide chain, and then we randomly do a mutation, mutate against some other amino acid. Then we do molecular dynamic simulations to really sample the bound conformations extensively. So not one single conformation, but many conformations from the molecular dynamics. And then we score these conformations using a consensus score that is determined from multiple docking programs. Finally, we accept or reject the new mutation if the consensus score is approved. And we do this many times in a Monte Carlo style algorithm. And so this is an example of how the design evolves. So the green amino acids are the ones that are accepted. The red ones are the ones that are rejected. And we have multiple scoring functions. And the idea is to minimize these scoring functions in order to have a statistically reliable score or average consensus score that tells us that this new computationally evolved sequence has a much likely higher affinity to bind to this receptor. So we worked a lot on how to do the mutation efficiently, how to place the side chain efficiently to sample quicker MD simulations. We've also worked on the consensus methodology. So what are the best scoring functions to use and what is the best way of combining these scoring functions to be able to have a statistically reliable, say, vote for the goodness of a peptide binding to the MHC class 2. And we've also made this computational protocol that we called PARSE, and in Colombian Spanish means friend, for general computational design. So now I want to talk to you about an application. The idea is this is a proof of principle, a question that we asked ourselves. So if we start with a peptide with known affinity, can we apply our computational protocol, our computational pipeline, and design a peptide that has a higher affinity than the original one? So one of the key issues was how to do the mutation. So we had the first strategy, which is basically random position in the peptide, random amino acid, but there were also other strategies that we used. So for example, reduce the hydrophobicity of the peptide chain, or reduce the number of synthesis rules of the peptide violated, because we're going to do experiments. And so in the end, basically here what I am graphing is our computational protocol designed around 100 new peptide sequences that in principle are better than the starting protein, the starting sequence. And then we are graphing here the number of synthesis rules violated and the hydrophobicity scale of these new sequences that we designed. And what we see, and in order to do experiments, is that we want peptides, new peptides that are not so hydrophobic, so that have a low hydrophobicity scale and that do not violate a lot of synthesis rules, because we know they're going to be much more difficult to characterize for the experimentalists. So we selected out of the 122 that had the highest scores and also conserved these properties. And we ran long molecular dynamic simulations around 200 nanoseconds per peptide. And then out of these, we selected 17 for experiments. So the experimental assay that we're doing is a competitive binding assay in which you have your MHC receptor, your peptides, and then if a peptide binds to the receptor, there is a fluorescent signal that tells you basically the intensity of the binding. So this is a proxy to the affinity of the peptide. And so these intensities can be scored. And here in purple is the reference, the starting peptide. And then you can see that we managed to design two peptides that had a higher score, a higher affinity than the original peptide. So basically what our algorithm tells us is that we can design peptides. Our algorithm is able to produce peptides that bind with higher affinity. And in particular, we've noticed that the sequences here are not so different from the original peptides. So a couple of mutations are enough. So now the main motivation of our work is that some vaccine trials work for certain populations, but fail for others. And so together with our experimental collaborators from the University of Sao Paulo, Daniela Santoro Rosa and Victoria Lunarelli, we went to design a panallele binding peptide. So basically a peptide that binds well to multiple alleles of the MHC class 2. And so, so basically here we have our parallel panallele binding design protocol in which we start with an epitope from Plasmodium vivax, and then we propose a mutation, and then we run molecular dynamic simulations, we score these conformations, and now we're going to accept or reject, depending if it's better for the majority of alleles. So basically, we don't want peptides that bind very well to just one, but what we want to create is this universal epitope that binds well to the majority of variants of the MHC class 2. And so we had several mutation strategies, so some in the Franklin region, some that were in contact with non-conserved amino acids of the receptor. And we designed around 44 new peptides. And here we're also plotting the hydrophobicity scale. So we want peptides with low hydrophobicity, low synthesis rules violated. And so we selected these for experiments. And so here I'm showing this cumulative binding score, which is used exactly in the same way as the experiment that I previously showed to you, but now we have the multiple alleles. So each color is a different MHC class 2 variant. And so the dotted line is the reference peptide, the starting peptide. And what we can see is that we can improve the affinity in 4 out of 17 trials. So this is from the drug screening perspective, this is a very high success rate. So I just want to finalize that our computational protocol has been proven efficient towards improving the affinity of peptides towards protein targets. And this is the first ab initio design that works for MHC class 2 and in particular for MHC class 2 panallele binding towards the design of universal epitopes and universal vaccines. So I just want to finalize thanking the person that did all the work, Rodrigo, my PhD student, and my collaborators, some of which are there in Brazil. And I also want to advertise that we have postdoc positions in a new group that I've started at the Flatiron Institute. So if you have students that are looking for postdocs, please write me. And thank you very much.
Okay, great talk. Thanks, Pilar, for your participation. Okay, so the next speaker is supposed to be Pablo Danz from the Universidade de la República in Uruguay, but Pablo asked me to play his video that he sent because apparently he's on travel. So let's see if I can make it work. 1 second. I'll share the screen. Let's advance it. Certo. Okay, share. Can you see the slides, Teresa?
Not yet.
Okay. Let me try again.
Yeah, I think it's coming. Yeah.
And let's see if the audio works as well.
Hi, everybody. So I want to start thanking the organizer for this wonderful satellite meeting.
Is it working?
I think the sound is very, we can't hear that well.
Sound?
Yeah, well.
So we are, as I told you, we are located in the Senur Litoral Norte. Actually, we are located in the Salto, in the city Salto, in the north of Uruguay. So we have the main headquarters at the Department of Ciencias Biológicas, the Universidad de la República. And we have a secondary office in the Instituto Pasteur of Montevideo, in the unit of bioinformatics. So we have to travel 500 kilometers once every 2 weeks, but it's okay. And well, you see there we have our web page. So if you want more information about our group, which is new in the region, you can get it there.
Yeah, I'm not seeing the video.
I was able to build this new group in 2019, thanks to the teachers I had in the past, and several of them are here today. So I want to say special thanks to them. I'm talking about Laura Coitinho, where I did I learned QM. I did my PhD there. I learned QM and QMMM methods. Then I also worked several years with Darío Estrin, where I continue learning about QMMM, and I start doing MD simulation, atomistic one. Then I moved to the group as postdoc, to the group of Sergio Pantano, where I did MD simulation, and I start working in the world of coarse graining biomolecules. Then I moved to the University of Farmacia in University of Barcelona, the Faculty of Farmacia in University of Barcelona with Javier Luque, where I continued working on MD, and then I also learned a lot about docking algorithms. And then I moved to the group of Modesto Orozco, where I've been working for 10 years. And there I did a lot of QM, MD, and coarse graining. Everything focuses to nucleic acid. So actually our group is devoted to study nucleic acid, and we are doing that with a multi-scale perspective. So we are working in multi-scale modeling and simulation of nucleic acid. We are working on DNA using all the possible methodologies, going from QM, QMMM, molecular mechanics, hybrid, MM, CG methodologies, coarse grains, pure coarse grained approach, and also using polymer physics to represent chromatin and chromosomes and whole nucleus. So we are using all the possible tools, and we are trying to integrate them for DNA, but also for RNA. And you can see there is some recent reviews we have wrote about these topics in the last years. So as I was telling you, so we are a theoretical group focuses on nucleic acid. We are developing new tools like Verina 3D, which is a tool integrated to the R package, which is QNIT. It was created to be able to do data mining of structural databases. Then we also develop MIOS, which is an integrated imaging and modeling strategy to resolve gene folding and chromosome folding with nucleosome resolution. We also work a lot in new models. We start working in coarse-grained models and chromatin models. So, for example, we collaborate with the DNA coarse-grained model that's implemented in the CIRA force field. We also work which is connected to a molecular dynamic algorithm to sample the conformation of space. Then we also develop another coarse-grained models in the IRB. Also with base pair resolution, but connected to a Monte Carlo algorithm to sample the conformational space. Then we went one step further and built a chromatin model that integrate nucleosomes. That was published this year in Nature Communication. But we also work a lot in atomistic force field for nucleic acid. For example, we worked a lot of years. We are co-authors of PARM-BSC1, which is the state-of-the-art force field for nucleic acid. And we also work in modification, enhancements of RNA force field, atomistic RNA force field. Actually, we developed PARM-BSC0 OL3 kappa. We work on the kappa modification, which is a modification that is able to reproduce correctly the puckering of RNA. And obviously, we also work on the implementation of specialized database. We work on some approximation to be able to store, retrieve, and analyze on the fly trajectories coming from nucleic acid simulation. So we have a big Nassim portal, which you can do these kind of things. And we also work on other kind of databases that, as the one that we call mini ABC, where you can get information on the static and dynamic structural heterogeneities of bDNA. So these are the type of things we are doing in the group. Of course, we are developing all these things, but we are also working in application of all these theoretical models. And actually, I want to show you some results about the last thing we are working on, which is this MIOS integrated platform. So actually, MIOS is a new machine for integrative structural biology focused on 3D and 4D genome. MIOS 2.0 is able to take as input a lot of different experimental measurements like high C or micro C experiments. You can integrate localization coming from high resolution optical microscopy. You can also add information from MNA-seq type of experiment. And of course, you can also put you can integrate chromosome models, chromatin models, and also docking algorithms to be able to dock chromosome and generate a complete nucleus. All these things can go inside MIOS and be integrated there to generate 3D and 4D conformation of folded genes, folded chromosomes, and also the structure and dynamics of complete nucleus. So here you have some examples of simulations of the one in the top is the folding of a segment of chromosome 16, and you have a gene there in green. And in bottom left, you have the conformational sampling of chromosome 12 with GAPDH and NANOC genes in green. And at the right side, you have the simulation for a complete haploid yeast nucleus, where each of the colors are a chromosome and the black sphere are the centromeres. So these are the kind of things that we are able to simulate, integrating several sources of experiments and several sources of models to generate this kind of systems and results. So I want to thank you for your time and your attention. These are so we are a new group, only 2 years now working in Uruguay, but thanks to God, we have a lot of students. So the group now has 7 persons, so we are very happy for that. And here are also the founding agency, so thanks to them also. And yeah, see you next week in the IUPAB Congress. Thank you very much.
Okay. Thanks for the video, Pablo. Sorry for the glitches in the beginning, but I think it's went on most of it. The next speaker is going to be Ana Elijah Scott from the Federal University of ABC here in Brazil. She also asked me to play her video. Apparently she had to travel as well. So I'll try to make it work.
Hi, folks. I would like...
Oops. Yeah, There we go. Share screen. And now, there we go.
Hi, folks. I would like to thank you, Guilherme and SBBq, for the organization of this satellite meeting and the invitation. Thank you very much for this opportunity to talk about what we are doing in our lab. I will share my slide, my presentation now, and my screen. Okay, and good. Let's go to start. Then, okay. The idea of this short presentation is to discuss an example of apply hybrid methods to understand molecular mechanism involved with some disease and involved with large-scale events in molecular system. I'm from Federal University of ABC. UFABC is in Santo André city, is the Great São Paulo. This is one picture from UFABC, the campus of Santo André. Okay, what our lab have done in the last years? Our lab in the last 6 years has worked with hybrid methods to sampling the conformational space of macromolecules, especially proteins, to investigate some molecular mechanisms involved with diseases as diabetes or HIV or now COVID. And then we all the time we are looking for this kind of collective motions and use methods that combine local motions from molecular dynamics and collective motions coming from normal modes analysis and experimental data to trying to explain this molecular mechanism involved with this disease. Okay, we know that protein is not a static molecule. We cannot see the protein as a static picture, but we need to think that protein, they are entities, so dynamics, and they have a structural dynamic that is very important for the function and to interact with another molecules, small or big, or another proteins. Okay, then all the time when we try to investigate these systems, we are trying to look for this relation structure, influence the intrinsic dynamics, structural dynamics, and the functions of this protein. Okay, we know that the protein can have different motions from and with different biological times. Then you have, for example, hydrogen bound vibrations between this time is 0.1 or 1 picosecond. And if you want to see collective motions of domains, or you need to think about microseconds. Okay, then all the time our lab is looking for this picture that tells us that the structure is correlated with specificity, affinity, and activity. The function is and influence the function of the protein. The dynamics aspect or structural dynamic aspect is involved with conformational adjustment, allostatic transition, domain motions, diffusion of ligands. And then nowadays we know that we don't have just a simple correlation, sequence, structure, and function, but we can think about in a more complex correlation between sequence, structure, function, and structural dynamics. And it's very important to see these macromolecules not like a single static picture, but an ensemble of conformations. Okay, there are several experimental methods to study these motions. For example, we can study using crystallography or NMR or cryomicroscopy or spectroscopy or calorimetry. And there are several simulation theoretical approaches. We can simulate it with elastic network models or normal modes analysis using atoms, or combine these techniques with molecular dynamics or metadynamics or another kind of molecular dynamic. And okay, then we have a collection of hybrid methods, and these hybrid methods that combine these motions, collective motions, generated by normal modes analysis with atoms or by elastic network models, with a method that can look for the local motions and experimental data. We can look for these biological phenomena with more precision. Then we have different hybrid methods and different combinations. For example, we have MDNM, molecular dynamics with stated normal modes that combine free molecular dynamics with normal modes analysis with atoms, and state the atoms in a direction of the combinations of the modes. Or, for example, we can have the co-MD that combine elastic network models and minimization of energy and Monte Carlo and a simple dynamic, or the mode, for example, that combine molecular dynamic simulations and normal modes analysis with atoms. Then we have a collection of hybrid methods that they are becoming, in the last 10 years, very important to study molecular mechanism. I will show an example that we apply in our lab. This is we are studying the human dopamine transport, and we are and this molecule is a channel that is this channel transport the dopamine inside of the cell. Okay, there are several experimental works that they show that some different phosphorylation conditions in these sites can increase the dopamine rate, efflux and influx in the cell. Okay, then we are trying to look, but no, until this moment, nobody shows which phosphorylation change which motions and these motions influence which state of the dopamine we can obtain. For example, we have from several experimental and theoretical works, we can define 4 states for dopamine transport, 2 intermediate states and 1 open state and 1 closed state. And this work we try to apply the MDNM to show which motions, how the phosphorylation change the motions, and how these motions change the probability of the states and the transition between the states and the free energy. This paper was submitted to the journal Molecular Biology. Okay, then what we did, we did a model. We built a model for the human dopamine transport. After we studied this dopamine transport using MDNM with sampling the conformational space, and after we apply the library PyEMA to calculate the macro states and the free energy of the states. Then I will just show, the time is so short to discuss this, but just showing. Here we are studying 4 phosphorylation conditions, all sites described in the literature phosphorylated. no one site phosphorylated. We have the Serena 12 not phosphorylated, and the other sites phosphorylated. And here we have the Serena 333 not phosphorylated, and the other sites phosphorylated. And we can see we sampled with using MGNN, and we can see that the distribution of RMSD change with this phosphorylation. Okay, this is obvious. But if you look to define these 4 states, we use
Okay, so I guess that was the end of Analisia's video. Apparently, she sent me the wrong version, but anyway, we move to the next speaker, who's going to be José Rafael Bourdin from the Federal University of Pelotas in Brazil. José, please share your slides and José, I think you are unmuted.
Are you listening now?
Yes, we
Hello?
can. Yeah, yeah.
No, okay, okay. Just 1 second. So,
but it's a black screen.
okay, I put this to share my screen. Can you see it?
Okay, now
no?
we can.
We can? Okay, perfect. Thank you. So thank you very much, Guilherme, for bringing this meeting, and it's really nice to see everyone, even that we are not online, or sorry, that we are online, we are not live. So I'll talk a little bit about a recent work that we did in my group. So it's called phase classification using neural networks. It's a little bit different from the other works because I'm a physicist working more physical problems, let's just say. So the problem is basically try to see or try to find the phase diagrams of complex systems. Obviously, this is a huge task because find the phase transitions and find the phase regions, not an easy task. And usually it involves a lot of calculations. For instance, calculation from some thermodynamic parameters like this one. So using these parameters and how these response functions fluctuate near the phase transitions, we can build the coexistence line and try to find some critical points. So this was our problem, how to study a system with a lot of phases as water. So water has more than 24 of solid phases, and also water has 2 liquid phases. And this liquid phase coexistence ends in a liquid critical point. And another huge problem is that we cannot reach this region by experiments, because usually when you try to cool down water up to this region, they crystallize to some amorphous phase. So we use computer simulations to try to understand this kind of system, not always using like a real model of water or atomic model of water, but some water-like fluids that also have a lot of solid phases. For instance, this model, we observe a BCC solid and HCP solid and an amorphous solid. Also have 2 liquid phases and with a coexistence region between this low density liquid and a high density liquid. And this coexistence ends in a critical point. And from this critical point, you have this widow line that split the region where we have a LDL-like behavior or a HDL-like behavior. And we was interested to see how some solutes will affect this behavior. So we start from one of the simplest solutes, that is alcohols. So we start with small chain alcohols like methanol, ethanol, and propanol. So we evaluate all these phase diagrams. So was 10 phase diagrams using classical molecular simulations. And obviously, if you did, this did a lot of work because you have to evaluate a lot of thermodynamic response functions, also structural properties and dynamical properties. Then we ask ourselves, okay, how can we do this easier? And nowadays, do something easier also, not quite so easy, is to use machine learning. So machine learning is a useful tool when you need to find patterns and or better, when you have to find distinct patterns. We can ask for a machine to learn to say if some picture is from a cat or from a dog, or in our case, we can ask for the machine to answer the question about in which phase our system is. And also, we want to do this really fast. So instead of use all the trajectory file from our simulations, we decided to pick only the last snapshot. So it's only the last configuration that we use to evaluate a series of orientational parameters. So they basically say how the system is structured. So we can change this this L for 3, 4, 12, and try to see what is the structural arrangement of the system. Obviously, this is not completely new. For instance, something like this was already applied by Martel and co-workers to study chip for P water and to see when you have the amorphous density phase or low density amorphous phase or a high density amorphous phase. And also, this was applied by Bottini to when you have a mixture of two colloids. So we have a mixture because it's water and alcohol, and we want to see what happens in this region of water when we have a lot of phases coexistence. So we add some extra that was this last term, where basically we try to include not only the first or second neighbors when you have your particles, you have your first neighbor or second neighbors. With this, we can include some information about the third neighbors. And why is this relevant? Because when you go for transitions like for the amorphous phase to the high density liquid, the structural changes are really small. You don't see some huge difference in your, for instance, in your G of R, as you can see when you go for BCC phase to the HCP and then for the amorphous phase. Just forgot to say, all this calculation was done using the Freud package. So that package is implemented in Python. Also, our neural network was implemented in Python. So we basically evaluate all these parameters for our system and then use this to feed our machine. And then after some magic tricks, the machine answers which is the phase. Okay, so we use supervised learning. Our idea is to go now for unsupervised learning, but we just pick 10 random configurations from 3 phase diagrams to train our neural network. So 2 points from each one of the regions, so the BCC phase, the HCP phase, or the amorphous phase, 2 points from the low density liquid, 2 points from the high density liquids. And we take this from 3 different phase diagrams, both with the alcohol fraction of 0.1. Here is methanol, ethanol, and propanol. So then we train our machine with all these parameters and ask machine, okay, can you see which phase is which? So we basically evaluate how much particles in our system. The total number was 1,000 molecules. how much of these molecules are in one phase or another. So we have the phase 1, BCC, phase 2, HTP, phase 3 is the amorphous solid, LDL of phase 4, HDL of phase 5. And then the machine has to say if the picture is a cat, is a dog, or is something between cat and a dog. So for instance, here we have almost everyone in the HDL phase, but then 10 particles are in the LDL phase. Here, everyone is on the BCC phase. So then we can map in the which phase each particle is, and use this to make our phase diagram. Obviously, if in this case, once most of the particles are in the HDL phase, the machine say, okay, this phase is HDL. And this was implemented using TensorFlow and Keras. Everything was implemented in Python. For more details, we can see our archive. The paper is submitted. Hopefully, will be published soon. So the good news is that our machine works. So the gray dots are the thermodynamic response functions. They, how can I say, they split the region in the phase diagrams, the region for each phase, and our machine works really, really well. Obviously, the machine only uses one snapshot, so it's really, really fast to get this kind of phase diagram. So, and also, obviously, to find the precise place of the transition, we need to do thermodynamic analysis, let's say like this, but we can have an idea where are the transitions and use this. I just have to run because I have less than 1 minute. And this works for mixtures. And also, including the extra neighbor, we can do a better mapping, just say what's the difference. Here we have the original neuron network. And as you can see in all the high-density liquid phase, you have this kind of amorphous invasions, or you have some particles in the amorphous phase, while here you have many points where we can split really well between HDL and amorphous phase, and this is a huge problem when we are trying to study supercooled liquids. Also, we can do these analysis populations to see what kind of transition you have. For instance, here you have some kind of first order phase transition when everyone changed from one phase to another. Let me just, and here also here and here. So here, even here, there is some 2 liquid phases. We can see precisely the transition from low density liquid to high density liquid, and we move for another temperature above the critical temperature. You can see this is smooth phase transition when you have the domain of the low density liquid at low pressure and a kind of domain of high density liquid at high pressure. And also you can see the transition from the amorphous phase to the HDL phase. So concluding, this the machine works. We can predict the phase diagrams. And next we are now building another package to analyze trajectory and try to prove our structural analysis for 2D systems. So all this work was done mainly by Vinicius. He was a master student here in Pelotas. Now he is in the Netherlands doing his PhD. So thanks to our government, we lost a great student. Also, we have Murilo did part of the works. So thank you very much again, Guilherme and everyone, the people who gave me money. And any questions, please send an email or don't hesitate to ask.
Okay, thanks a lot, José, for your talk and participation. Okay, so now we're going to the last talk for the session, for the first session. And it's going to be from Teresa Soares, from the University of São Paulo, here in Brazil. I guess, Teresa, can you share your video? We are going to change a little bit. You know, she's not giving a scientific talk, but here we go.
Good afternoon, everyone. It feels good to meet everybody again. I hope the next round we will be able to meet physically. So I would like to start thanking Guilherme for putting together this nice satellite meeting. And I'm also indebted to you who remain until the end of this session. I will try to be brief so that we can have enough time for questions and discussions. So today I will bring news on one upcoming special issue with JCAM that I think will be of interest for the SAIMS community, and I would like also to chat about JCAM new policies on data sharing and reproducibility of results. In the flow between those subjects, I would like to quickly go over the types of manuscripts accepted by JCAM and how to engage more colleagues in peer review. So a few words on our impact factor for 2020, which is now 4.956, with a total of over 24,000 citations. JCAM impact factor has been continuously rising in the last years, and we expect this trend to continue in 2021, of course, with the help of JCAM authors and reviewers. Our 5-year impact factor has also been increasing, which shows that the research published in JCAM has a long-term impact. I believe that much of JCAM's success derives from our desire to be inclusive of exceptional research produced worldwide. We maintain an international board of editors, which is also gender balanced. We keep a dynamic advisory board with a broad range of expertise, which we can count on. And nicely, last year we had Ariane Nunes Alves from Brazil and Matias Machado from Uruguay invited to compose our early career advisory board. They are doing a superb job and opening the way to increase the participation of more South American, Latin American researchers in our advisory board. This is something I'm deeply committed to promote during my editorial term, and I cannot acknowledge enough the support I have received from the SAIMS community, whether as authors or peer reviewers. So now on the coming JCAM special issue, next year we will have the second special issue on women in computational chemistry. This was an extremely well received special issue by the community with the largest number of submissions in JCAM history. Actually, the second largest number of contributions received for a special issue was the one on molecular simulation in Latin America, and then thanks to the support of the SAIMS community. But yeah, so now JCAM is launching a new call for papers for the special issue that will be entitled Advancing Women in Chemistry, which aims to address and close the gender gap in chemical science. This special issue is meant to highlight the fact that science is for everyone, for women, people of color, disabilities, minorities, transgenders, and they can all make a difference. That's what we believe. And in this spirit of inclusion, we invite scientists from around the globe to submit manuscripts focused on a combination of experimental work and chemical information or modeling or just theoretical work. We welcome all types of manuscripts which are consistent with the scope of JCAM. Most importantly, we welcome all authors irrespective of gender. However, for the manuscript to be considered for publication in this special issue, the authors should provide a statement that they are committed to supporting and advancing women in chemistry. The editorial for the special issue has just been accepted for publication this week and should be available as ASAP publication on the JCAM website. So the deadline for submissions is December the 15th. And I hope we are going to have some nice, great work from the SAIMS community being showcased in our special issue. So I would like also to briefly discuss the manuscript types accepted by JCAM. Although this information is available on our website, I think there are some categories of manuscripts that I would like to highlight with the community here. So besides the well-known types, such as research article, letters, and reviews, JCAM has 3 categories of manuscripts that are somehow special. One of them is the viewpoints, which are general commentaries on current issues in the computational chemistry field, or scholarly comment on papers appearing in JCAM. Viewpoints will be rigorously reviewed by at least 2 JCAM editors for content and the importance of the topic. Following the editorial review procedure, select articles will be sent out for peer review for final assessment according to editor's discretion. The second type is the perspectives, which are interpretive accounts on subjects of current interest to the JCAM community. This series is intended to be a forum for experts to present their perspectives on emerging or active areas of research. So per submissions, inquiries should always contain a brief summary of the proposed topic and be addressed to the editor-in-chief office. And the third type of manuscript, which I think is something really special, and I think we are at least one of the few journals that have such a type of manuscript, that accept such a type of manuscript. So those are the application notes, and those are informative peer-reviewed reports on novel software packages, database, and web servers described in a succinct manner, so it should be below 5,000 words. The name of the application being described should be clearly stated in the manuscript title, and the scientific, technical, or other usability advancements of the software should be clearly described. As far as possible, the software packages and web servers should be operating system agnostic, running on different platforms. And all the submitted manuscripts will be reviewed by an editor prior to being sent out for peer review. It's also important to remind authors that the software should be generally available for evaluation or purchasing at the time of publication for academic and commercial use. And the software must be made available for testing by reviewers to address specific data or claims in the manuscript while preserving reviewer anonymity. And that brings me to the next topic, which is JCAM new policies on data sharing and reproducibility of scientific data. So I think we all understand the importance of data and method sharing to ensure the reproducibility of published results and observations. This has been the topic of intense discussion within the computational chemistry and biology research community. Scripts published in JCAM since January 1st, 2021, authors are required to provide information about the data and software described and used in their manuscript in a separate data and software availability section at the end of the manuscript. If the authors have reasons not to share data and software freely, they should state the reason. For instance, data is considered to be proprietary by the organization, but then the authors should explain why their work has unique scientific value. I encourage authors to read the editorial before the next submission to JCAM and to familiarize themselves with the new requirements. So I think we all understand that peer review exists to validate academic work. It helps to improve the quality of published research years. Several SAIMS members have provided invaluable assistance acting as reviewers for JCAM. I know this is hard, anonymous work that perhaps receives less public recognition than it deserves, though nowadays reviewers can receive credit for their work via Publons or CID. However, sometimes, perhaps often, we are overloaded with work and cannot take review assignments. In such case, there are some helpful actions that I would like to suggest to the community. So the very first thing that could be done if you cannot take a review assignment, a peer review assignment, you could suggest the name of one or two colleagues who are experts in the subject, and if possible within your country or Latin American countries. Another possibility is that you could mentor a young research assistant or a postdoctoral fellow throughout the process. Of course, do not forget to inform the editor handling the submission about this arrangement, and keep your mentoring strictly to the technical aspects of peer reviewing, allowing for the mentee to exercise critical assessment of the merits and flaws of the work in consideration. I strongly encourage less experienced but also more experienced reviewers to take the course. It is very helpful in defining a systematic approach to peer review with 6 basic key steps that will make the process of reviewing scientific manuscripts more streamlined and consistent. And with that, I'd like to thank you for your time, and thank you also, Guilherme, for making space within the satellite meeting to talk a bit about the news from JCAM.
Okay. Thanks, Teresa, for the talk about the journal. I think I would like to invite all the panelists to join in this discussion about their work, and to reply questions from the participants, from the audience, and from ourselves as well. So it's open for questions, please. Open your microphone and speak up. For the participants, you can send questions by the Q&A tab, and I can read them if you point to a particular panelist. Yes, please, Mauricio. You raising your hands?
Mauricio is muted.
Well, you should be able to unmute yourself. Let's try.
Okay.
Yes.
I think Guilherme was making sure that I do not ask too many questions.
No, I haven't done anything.
I have a question for Esteban, and you mentioned that your intention is to do a better charge model for high throughput methods, but I think you answer here quickly on the shot that your chargers are host based. So how complicated or difficult it is to tune for every host compared to, for example, a usual high throughput, which you have unpolarized charges. Yeah, you're muted also.
What is it?
Okay, now I can speak. Thanks.
Okay.
So thanks for the question. Yes, that's a very good question. Actually, I had no time to go into details, but we also checked basically to take host agnostic charges. That means that we derive the charges in, for example, Echo solution, and we apply them to different hosts. and the results are not as good, but they are still better than the standard AM1 BCC charges. So the reason they are not as good is because for some guests, polarization effects are important and contribute to binding affinity. So if you need them to be included, you have to derive environment-specific charges, but sometimes you don't have to. Depends a little bit on the guests. So I don't have like a general answer, but also if you take charges which are not derived for each host, they are still, and then you can basically apply them for different hosts. They're still better than the other ones. I don't know if that answers your question.
Mm-hmm. Thank you.
Okay. I hope everyone is able to open his mic or his or her microphone and speak. Teresa, there was a question in the Q&A about your talk.
I think I replied that, right? It was Matthias. So
okay.
that I have replied, yeah. So about this new policy on data reproducibility. And I guess this is something that we all agree about, right? We all think that is good to make things transparent, that, you know, everything is made freely available. But of course, there are also authors that are in the industry, and sometimes this is just not as easy. And of course, this has to be taken into account. So the idea is that we would like to have those as much as possible software available, at least for testing by reviewers. And then, of course, we also need to count on reviewers to request that they have access to test those things. So I think this is what I sort of said in a few words to Matthias.
I see Munir Iskaf has raised his hands. Munir.
Thank you, Guilherme. It's so nice to see so many friends. It's wonderful. Thank you very much. It's a great pleasure to see you all. Everybody looking healthy. It's very nice. I have one quick question regarding, not question, but actually a comment regarding data sharing and you know, making available results. Universities, especially here in São Paulo, they were actually, this movement was taken ahead by USP and also by Unicamp and other universities that have created institutionalized data repositories, where you actually you are complied to deposit your data, but not just your data, but your codes, and it has been working really nice. And there is one additional piece of information is that the data that is shared there, codes that are shared there, they are all connected and they are indexed by Google. So your citations to your work increased considerably because the data shared can be individually cited. So you get citation on the data if your data is on these institutional repositories. So I just want to make sure that everybody remembers that and knows that and use that. That's all. Thank you very much.
Munir, can I ask something about that?
Yeah.
Is this an open initiative for all São Paulo universities or just for the state system?
all São Paulo universities, including UFABC, even the private ones, and this movement is capitaneado by FAPESP. So everybody is connected through FAPESP in this data repository.
Very good. I didn't know about that.
If Muni allows me, and actually this can be, we request that you sort of offer a provided address where the code is deposited, and that can also be the FAPESP supported or USP or Unicamp connected database. So could be GitHub, but could be also an address like this. So this is, it all goes well together.
I have a question for.
I think I also have a question.
Sorry.
Yes, it's for Teresa, and Teresa, it's necessary to make available the code of the software, or it's sufficient, for example, like Doctor, to make available the software by a web server?
Yeah, I think that what we mean is that you don't have to make the line by line code available, but the code must be in a place that can be tested, can be used, and you have to describe enough that it can be reproduced. But if the code is an open source code, of course, then you don't have to describe it line by line, like you don't have to publish the code for that.
But my question is, if I didn't want to make available all the code, but I only make available the use of the code by a web server, is it sufficient for the JSAIM or not? Or I have to make available a free software, because there are many I'm talking about the Doctor, because I'm in an institution from the Minister of Scientific from Brazil, and I have some concerns with the minister, the director, and but the software is available by a web server. It's sufficient for the JSAIM, or I have to put the code of the software in the public repository?
No, I think if you first of all, everything can be explained and justified. This is the very first thing, and of course, it's up to the editor-in-chief to give his view whether this is justified or not. But I think it's in this case is a code that is available on a place that can be accessed continuously by users. It's open source, it's free to use. So in a case like this, let's say if you submit a paper and you describe the application of Doctor and you make inputs, everything available, any person can just go to this web server, use the code and reproduce the data. I think the main point here is just to ensure that we are publishing material that can be fully reproduced. I think that's the main thing. So in a case like this, my understanding is that the code is available and therefore it can be used by users or reviewers to reproduce your data.
Thank you very much.
Mauricio, you're going to say something, I guess.
So my question is for Diniz. On your very nice work, and you do the replica exchange, did the system switch between the two states, the unactivated and activated, or did you see, I mean, those two states are very well defined, or you see a lot of stuff in between this transformation for the GPCRs?
Well, the first simulation with the constitutively active movement, we observed the full transition to the active state. Well, actually, it's a little bit hard to say it's really a full transition because the receptor is bound to the G protein. So the available structure in the active state has a nanobody mimicking the G protein to keep it open like that. And the other structure that was released in 2018 has the G protein in place. So in my simulations up to now, we don't have neither the nanobody nor the G protein yet. But with the constitutively active mutant, we saw this change in conformation, which is very typical to the activation of the receptor. And when I did it to the wild type protein, we had some conformational changes that were not enough to say it's a really active state, but it's in the middle of the process, of course. And also these receptors, they don't have just active and inactive. These are the structures that were captured on the X-ray crystallography or the CRIV-EM experiments. But there are several conformations in between that we can up to now only access through molecular dynamics. Okay.
Yep, thank you.
Okay, further questions? Sergio, please.
Yes, I have several ones. Ernesto, are you there? Because I'm not seeing you. Or actually, it's a general question if you want. Ernesto is apparently.
Yeah, Ernesto sent me a message. He apparently had to leave for a little bit, but he said he would be back. I'm not sure.
He was not well, I guess, he said.
Oh. Okay, so I have another one for Bruno. Bruno, have you used the what's called GMAC to test for the robustness of force fields? I mean, what happens if you change like 10% of the energy barrier or something like that? Would the force field be still as good or as bad, if you want, as it was? I mean, have you played with kind of small tweakings and just to push it to the edge? Or sorry, let me reformulate. What happens if you select with GMAC a set of non-optimal solutions? So not the best one, but kind of the near ones to that.
Hi, Sergio. So that's a good question. Yeah. This is an important issue because when you have multi-objective functions to optimize, for example, we want to optimize the force field aiming at several properties. Okay, so in the beginning of Gromos, Wilfred van Gooseren decided, okay, I will target the density and the heat of vaporization of the liquids. So he had a two objective function and several parameters in the force field to optimize. Now we are adding more target properties in the optimization, and then the problem gets more difficult because with the same set of parameters to vary, let's say in the example that I've shown, we only had two parameters, the C6 and the C12 of one of the atom types, and we were optimizing this pair of parameters to reproduce three different properties. The density, the heat of vaporization, and the surface tension. So we had more properties than parameters. And we ended up in this Pareto scenario where you don't have an optimal, a clear optimal solution, but you have to make choices. Because, for example, we can have a force field that is perfect for reproducing the density and the heat of vaporization, but will fail for reproducing the surface tension, or we can choose a solution that's more ideal for reproducing the surface tension, but of course we will have some drawbacks in terms of density and heat of vaporization. So what we are doing is that we characterize this Pareto front, and then we are able to choose from all the set of optimal solutions which are the solutions that we actually want for the force field. And the way that we are doing that is that we are validating the set of solutions. For example, let's take the solution that best reproduces the density and the heat of vaporization, and let's analyze what happens in the simulation of other properties to see which of the solutions of the Pareto front is mostly transferable for reproducing other liquid properties. For example, we can take the diffusion coefficient as a validation property, for example, and say, okay, let's take the different solutions that we get in the Pareto front. We take the solution, for example, the one that of the best density, the one of the best heat of vaporization, but let's take all these solutions now and calculate other properties for which the force field was not optimized for. And then with these additional properties, we can restrain the Pareto region to select, pinpoint better which force field I want. I want a force field that's able to reproduce all these three properties, but also some other properties that are there, or which of the solutions is more transferable to other properties. So that's the idea of it. But of course, sometimes it's impossible to find a set of parameters that will give optimal solutions in terms of all the properties that you select, especially if you have less variables in the parameterization compared to the number of targets in the parameterization. I don't know if I was clear, but this is the strategy that we are using.
Thanks.
Welcome.
Okay, Nina Pastor, please.
Yeah, hi. I have a question for Hugo Verli. Regarding the parameterization for sugars, how does this your particular force field compare to, say, Charm 36? I'm thinking about some recent simulations from Romy Amaro's group, where they simulated the spike protein with all its glorious oligosaccharides. Is this science fiction? Can it be taken seriously? If you're looking for, say, another cool epitope to try to develop an antibody, should we care about it? Or it's still glycans are a mess and that's the way it's going to be?
As far as I know, we can see the newest version of FLSA, charm, glycan, and gromos behave quite well in the reproduction of carbohydrate geometries. The problem is this delicate conformations that the force fields are parameterized for the best conformation in solution, the most abundant. That usually is okay. But if you want to see some delicate phenomenon, enzyme catalysis or some carbohydrate protein recognition, then you don't have the necessarily best answer. For example, some residues glycan predict as the opposite conformation. So you have some specific difference, but in the most general residues for glycan chains, it should be okay. And the models, I think, should be very well, very good.
Okay. So structure and dynamics, fine.
I think so, and I think they are the easiest way to access carbohydrate information, structure information, much better than you have for proteins. I think the accuracy is better for carbohydrates than they are for proteins, for example, in the most situations.
Thank you.
Okay, Esteban, please.
Yes, I had not a question. I had a comment for Bruno actually, with regard to the question of Sergio. And I think it might be because for example, for C6 coefficients, there are physical values basically from which you from isolated atoms basically. So basically, it's not that you are completely blind in parameter space. So it might be a good idea to restrict the values on your Pareto front to have a physical meaning, because a physical, how far you can go for physical, but that I think will restrict the range, and then basically you will also reach more transferability basically. And that's actually, I think, what most of the people who are doing force field development basically do. They try basically to guess something which has some physical chemical intuition and which is transferable, because if not, I think you described very well. I mean, you have only some values you know, and you have a lot of parameters you don't. So it's important. It's impossible to get them all right. It was just a comment.
No, that's a good comment. one of the things that I don't like so much about this physical meaning of the C6 parameter is that the C6 parameter has a physical meaning, but it's a physical meaning for the isolated atom in vacuum. And the atom is not in vacuum and the atom is in the molecule. So it's highly affected by the coupling with the other atoms in the molecule. But yeah, also the range of change, the percentage changes that we are talking about here is very, very tiny. It's just small perturbations of the parameter. So we are not talking about changes of more than 5%. It's really, really, really small changes, like less than 1% sometimes. So you clearly see in the graph plots that I shown, when you diverge a little bit too much from the optimal solution, it's very steep. It goes to regions of the parameter space that are completely unimportant. So the code, the methodology is good for small changes in the parameter. If you go to high changes, it's not working. So, but yeah, that we don't have to worry so much about restricting the parameter space because of that. In this regard, it never goes beyond a very huge difference in terms of parameter space.
Okay, and I think Lohan, you've got something to say. There was a question or a comment to you.
Yes, I have two questions to Pilar Cosio. Very good work. I have, when you optimize your peptides inside the MHC, can you use only docking, letting the side chains of the protein flexible also, and not only the side chains of the peptides, or you have to use molecular dynamics obligatory? It's the first question. And the second question is, because if you can do this, it will be more fast, it will be faster. And the second question is, you told that you use a random mutations in the peptide. You don't think that using a meteoristic, like a genetic algorithm to drive this mutation will be a better choice?
Yes, thank you so much. These are great questions. So in certain cases, we've seen that molecular dynamics, we do explicit solvent molecular dynamics, helps us a lot in obtaining conformations that are in equilibrium. So the problem in many cases about docking is that you have some type of outlier conformation that is scored. And so we've seen that having explicit solvent molecular dynamics takes away these outliers that can be like scored very highly with the docking scoring functions, but that somehow like have very short distances or are unphysical. So in most of the cases, we've seen that MD helps. But I think it also depends on the particular system that you're interested in. So this MHC class 2, the peptide is very flexible. And in this case, it's just like rotation of side chains or backbone movements is not enough to capture the flexibility. And for that reason, we need molecular dynamics. But if you have a very small peptide, maybe just docking is enough. And we need many trajectories, many conformations to be able to average these and score. So we're not using one, but many. And in regards to your second question, yes. So doing random mutations is not optimal. And in fact, we've used bioinformatic tools that tells us probabilities of amino acids to be at certain positions of the peptide, and use these to predict better mutations. So yes. Not random. So all of these things helped.
Because molecular dynamics is very expensive, but you can imagine, for example, a genetic algorithm where you use the docking methodology as a surrogate model. and only use molecular dynamics when you found some peptides that you think that are really good. And by this way, I think you can improve your search and have use molecular dynamics only for the best case that you are investigating.
Yes, more like a funnel-like strategy. Yes, might be more optimal computationally.
Thank you very much.
Thank you.
Okay, there were a couple of questions from the participants, but it's still only one now from Matias Machado. It's more like a comment saying that to Laurent that there are published extensions to Tinker in JCIM. So Sergio, if you can ask your question. And I think just one thing, I think that's going to be the last one because we need to have a break before the next session.
Absolutely.
And, yes, please.
Yes, that was I mean, it was pretty much the same, or actually it's slightly different for Pilar. Hola, Pilar. So there was a question in the Q&A and about whether you can implement it in like twice, I mean, 2 consecutive mutations in the same Monte Carlo, which of course will kind of explode the probabilities. But in this particular case, since the peptide is flat into the crevice of the histocompatibility complex, is it the fuse actually, because you're not expecting to have so many interactions? Perhaps you could do mutations at most twice at the time, but more than that, they shouldn't be so much connected. Am I right?
Yes. So yes, I think so. So because it's extended, maybe it's not. It won't be so cooperative. It's likely that you imagine mutations that have some type of interaction between each other that are the ones that will be favored. But in many other cases where you have the peptide interacting more with, like within a core or something, then I think it could be very useful.
All right. So thanks.
Guilherme, sorry, if I may very quickly.
Yeah, sure.
Just because maybe I was too harsh on Esteban, and now I think I get the point of one of the sides of the coin that I didn't overlooked when I answered him. Esteban, you are right that the starting point has to be physically meaningful also, because if you start from a gas, right, the gas has to be physically meaningful. And when I my example is starting from the Grom's force field that we already assume that has a good physical captures the physics of the model. So, and then we make small perturbations of the parameters. But of course, we should not go too much further away from these physically meaningful parameters. And in this regard, I only responded one part of your question. Thanks. That was a great question.
Problem at all.
All right. So I would like to collectively thank all the speakers for this first session and announce that that's over, and we're going to be back in about 10 or 12 minutes for the second session. And I'll see you in a bit. Thanks a lot.